Hello, I'm a professor at the Department of Animal Science at Laval University. I'm here to talk to you about a project which is called Improving the Fertility Genes of Cows. To situate the project in its context, it's important to present a few basic concepts since this is a project that deals with some data. You have to introduce the genome. I have to speak about this. The genome is something that is very big. We don't necessarily realize that, but actually cows have 60 chromosomes, 2 times 30, a series of chromosomes from the father and another from the mother. As all mammals, the size of the genome is similar. It's about 3 billion bases. It's very big. And regarding polymorphisms, that is what interests us. There are differences in the DNA sequence that we're interested in, mostly what we call SNPs. The SNPs are a substitution of one base by another base. Sometimes by these sequences of 4 letters, there's a substitution of one letter by another in a word. That words are written with 4 letters each. When we compare the DNA, sometimes we don't have the same letters at the same place. So these differences are what we call SNPs. We have to know also that the genomes are very large, but as a whole, they produce proteins. It's the reverse, actually. A large part of the genome is not a coding part of the genome. A large part of the genome contains, just like human genomes, about half of our genome is vestiges or remnants of regions that are repeated here and there and everywhere in the genome. Mammal genomes contain between 22 to 23,000 genes. It's not totally characterized, but it's somewhere around 22,000 genes. What are those genes? Actually, the genes are the codes that allow us to produce proteins. If you took all the coding regions side by side, they will produce all the proteins in all the cells, all the organs in a total individual. Basically, all this coding sequence would be about only 1 to 2% of the total genome. And that corresponds, if we take the length of the genome as being this black line here, that would correspond to the beginning here, to the handle. So in all our genome, there's a very little part that is dedicated to produce all the proteins and other products. When we speak about genomics, we speak about genotyping. Basically, we get the information corresponding to those nucleotides. Actually, we wonder, what nucleotides do I have? Is it an A or a G? And the genotyping platforms that have been developed for genomics have not been developed like people think, by basing ourselves on known information to associate sequences with animal performances. Actually, we chose sequences that were different between 2 parts of DNA at regular spaces everywhere in the genome. So those markers are not known as being associated to special characteristics of the animals. Basically, a way of writing a genome as a whole is a bit like a barcode, which you find on products at the store. So each animal received 2 copies of a genome, from 1 copy from the father and 1 copy from the mother. They might have received twice the same copy in certain places, and in other places, it could be 2 different copies. What we see here for the health characters and longevity characters we're interested in. We want to understand, is it good to have 2 copies, or is it better to have received 2 different copies? What is best? As this is what we see here. So there are 3 possible genetic combinations for each place in the genome, which could be bearers of 2 alleles, or we can have 2 blue alleles, homozygotes, for example. The heterozygote is the one who is bearer of each of the 2 alleles. In all genetic evaluation system, whether it be the traditional systems or the biogenomic system, it's a linear system is used. A linear system, how does it work? Well, basically, it takes it for granted that there's one of the genotypes, because in one of the type of homozygotes is let's say the red, reverse, those who have two blue alleles are the best. He's the bearer of one allele of each type. He's in the middle. Actually, he has a performance which is halfway between the other two. So is that situation valid for all trades? Basically, all situations that can exist do exist. Is it possible that sometimes there are bearers of two different alleles which are better than two homozygotes? Is it possible that the heterozygote animal is not really totally in the middle of the two, but a bit more like here, or maybe underneath? All possible cases exist, except that the mathematical models we use take it for granted that the system works linearly like this for everybody. That's what we call additive systems. So what we have to understand is when we make genetic and genomic evaluations currently, we calculate the effect of different parts of the genetic contribution, as what this is what we see here. Basically, that additive contribution is part of the traditional genomic and genetic evaluations. When we talk about dominance effects, we take it for granted that this is valued in a system of hybrid vigor. So an animal that will have two different alleles will have more vigor. There are other types of interactions that are complex interactions between different regions in the genome, in which we understand very poorly yet. And we don't take them into consideration because we can't estimate what their impact is. So we take it for granted that we can evaluate what we can calculate only. So only the additive contributions. I'll bring back this slide to show you that in an additive system, what it means that the bearer of twice the same allele, so the best of the two alleles, is the one that will perform best. If we select for all traits in an additive system, we could expect that all the genome would be pretty similar from the mother and from the father, because we selected individuals that had the best performance, and in those cases, it's the homozygotes that are the most efficient animals. So eventually, at term, we could believe that we could homogenize the genome by adding, like here, we added a pink allele on both sides, and here a black allele for a trait or a gene that is involved in fertility. So the question is it okay to do that? Does it work? Well, as I will show you later, we see that there is genetic progress, but maybe not for all traits. And what we see is that for genetic progress, we have to estimate what would be the contribution and the interaction, the additive interactions for all the performance. We can base ourselves on the hereditary aspect of traits. The heritability of traits is the capacity of transmitting this trait to the descendants. There are traits that are more easily transmissible than others, and technically, the contribution that is the most easily transmissible would come from the additive contribution. So that gives you a perspective about the proportion of additive contributions that exist for each trait. What you can see here, we'll take the Holstein as an example. Here you see for the production trait, what you see is that the contribution or the inheritability of trait is around 25 to 26, 28%. To give you an order of magnitude, we consider that everything that is a low inheritability will have something under 20. Between 20 and 40 is pretty much inheritable, and above 40 is extremely inheritable. In all species, these numbers don't change a lot. Traditionally, fertility traits have very low inheritability. And generally, it's very often under 10%, the inheritability values. Here we see the body conformation traits, the depth of the udder, for example, that are very strongly inheritable. Here we see stature, which is very, very inheritable, and the age. There are certain traits, such as the angle of the foot and others, that have very low inheritability. That gives you a good indication about how many generations it will take before getting a gain in at the herd level. The more the inheritability is low, the longer it will take. So the goal of genetic selection is to make the averages move and shift. Here we have an average, and when we select the best individuals, we make them reproduce. What we hope is that the descendants will have a different average and they will have progressed. So animals will be more productive on average. So the more there is great inheritability, the more that gain will be fast from one generation to another. The lower the inheritability, it will take more and more time to more and more generations before getting to the same point. We can see here the gains made over some 30 years. What's easy to see on the graph is that if we take the trait of health and fertility in red, there's been very little gain over the 30 years. Contrary to that, we see that production trait had very strong gains in the last 30 years in terms of confirmation or durability. We see that the gains were only marginal and sometimes a bit higher. But does the system work? What we see here is that the system works in terms of body conformation. We see that over 30 years, the animals have changed very significantly in the last 30 years in terms of their stature, in terms of the shape of the animals. They responded well to selection. All this was done with traditional selection. Now, the other question we can ask, have they reached a plateau or a ceiling? This is an extreme example where we see that animals can still produce very much. So we're not yet at a plateau. It means we aren't capped. We can still improve things. But the question is, are we working in the right way for the traits? If we look at the traits, as you know, traditionally, we didn't know what was in this black box, those genes. We didn't know what the genes, and that was okay. It still worked. Selection, we selected the best individuals, and the best individuals with the best performance are those that transmitted part of that gain to the following generation. Now, with genomics, as I said, we don't select for specific genes, but we select for a whole barcode, which is associated with high performance animals. It's not so different, but what changes is that we do genotyping today. So we describe the genome based on those SNPs that I talked about earlier. And so as I showed you, what is a SNP? If we compare two DNA sequences, what we realize is that it's a basis, which is a base that is different from one individual from another. That's what is a SNP, the single nucleotide polymorphism. The genotyping sequences address about 50,000 of those SNPs in all the genome, and that generates a barcode. And then we associate this barcode to a bigger bucket of milk. It means that more productive the cow is. When we compare performances, we compare this with the barcode. And when we have animals that we don't know what is the performance, like calves or even embryos, we can get the DNA of embryos, we can generate the barcode. And then by comparing the barcode, we can determine whether this animal has the potential of generating a big bucket of milk or a small bucket of milk. That's information which is available in the sense that here, as we can see in 2017, what was presented in Canada, we have about 2 million animals that have been genotyped. That is an incredible quantity of data. It's a database that is totally phenomenal. I'm coming here to the project. The current project comes from a first project we had done, which had been financed by Novalait, the CRIBIC, and NSERC. The goal was to determine, can we use genomics to try to improve the traits of health and fertility? Based on the results we had in the first project, The project that is currently ongoing is to increase the quantity of data in order to determine whether this pilot project on a small scale could be extended and expanded to the whole national herd of all cows in Canada. And the reason why we're interested in the health and fertility traits is because we know that there's a certain homogeneity in the population. We know in particular that this is referenced by the selection process, but influenced also by consanguinity. You know that consanguinity is something we see on the Holstein. The average is 8.7. If we take a breed, which is well known as having more consanguinity than others, the Canadian cow, for example, at 9.16, what is of concern is the fact that the annual growth is 0.25 for the Holstein. We see it well here on this graph. Here you see the growth of consanguinity since the 70s all the way to 2019, and we see that there have been gains that have been faster than others. These gains in the 90s are associated with the fifth Starbucks that were very much used. This is mostly Goldwyn, and here from 2009 is the arrival of Genomics, and we see that the gains, in the annual gains, we see that we're part of a very significant growth. Even in the last 2 to 3 years, I would say that there's been a shift and there's more and more consanguinity in breeding. So traditionally, there are stars that come to the fore. We see this when we compare the Holsteins and the Jerseys. On the Holstein, you see the 10 herds that have the most influence. But when you look at the number of descendants, we can see that we're in the millions. But for the Jersey cow, we're only in the hundreds of thousands in terms of paternal lines. Even if the Holstein population is very, very large, through selection, the genetics is smaller because there's a lot of craze about certain bulls, and those star bulls have a lot of impact on genealogy. So the idea is it better to have 2 identical alleles for the traits of health and fertility, or would it be better to have 2 different alleles? The working hypothesis is that genome homogenization is advantageous for some traits, but other traits, including health and fertility, could benefit from having 2 different alleles instead of always the same one. So the way we proceeded, we chose the best individuals for health and fertility, and we looked into the genome. We delved deeply to find if there were regions that were heterozygotes systematically for all individuals. We did find a few. We found about 800 regions in the genome, which is a small proportion of the genome as a whole. And we looked here at correlation with different traits. What you see here in red are the interesting correlations for the health and fertility traits. We also made sure that the targets we were choosing did not affect the other traits, the production traits. The idea is not to improve the health and fertility traits at the expense of production traits. So by targeting those regions that have that type of response, we were able to do the pilot project, and we tested it in a cow population in Alberta. That's a dairy farm of about 1,000 cows in lactation, and collaborating with Marcos Colazo in this collaboration. We worked to identify cows that had health problems and were sick, compared with cows that never had any health problems. So some had chronic problems, others had never had anything. And at the beginning, we didn't know what to expect. It was a pilot project, and we didn't know if we would identify the right regions. We didn't know if we could find those types of data from the cows. We were very happy to see that actually we have a trend here. Animals that are heterozygotes for the regions that we targeted have fewer health problems if they're heterozygotes in some specific regions. So the idea is to develop a plan. So if you have a cow, if you have the choice between 2 bulls, which of the 2 would be the most beneficial? So if we target the regions that we just identified, we will be able to say, what is the probability in this genotype that the descendant will have? And we can identify 1 bull in particular that will be better than the other. A tool was developed to allow us to take the genotype of cows, and then based on the known genotypes of various bulls, we can select the one that has the most chances of generating a descendant that will be heterozygote for the targeted regions. The projects that is currently ongoing, as I said, is to increase the pool of data. And right now we have access to a full set of data over 58,000 animals. So we have the production values, the performance values to rank the individuals as the better and the less good. And we have the associated genotypes. So we're currently compiling and structuring the data, which takes quite a lot of time. Then we will complete the genotypes because sometimes there are missing data, and then we'll test the models that have been developed in the pilot project, and we will try maybe to redefine this model with new data. Then we will develop the graphical interface, and eventually we want to transfer the tool to a Lactanet so that it becomes useful for your actual choices of bulls in the future. I want to thank here people involved in the project. Alexandra Carrier, who is here, then Alexandre Bastien here, and finally, Julien Prunier. And of course, all the financial partners and the funders without which we wouldn't be able to do the project, including Novalait, which has supported us for both projects. Thank you very much.